Hello, my name is Juan Santiago from Stanford University, and I'd like to present to you today an assay we've been developing for the detection of SARS-CoV-2, which is the virus that causes COVID-19. We use an electric field based purification, and we also use on-chip electric field based speed up of a CRISPR assay using isotophoresis. My collaborators in the med school are professors Benjamin Pinsky and Niaz Banaei. Ashwin Ramachandran, this is part of his PhD work. Diego is also a student in my group. Eesha, Malaya, and ChunHong Huang are postdocs in the team. Just to introduce you very briefly to CRISPR type diagnostic assays, you might be familiar with CRISPR as a gene editing, but a few years ago it was shown that it could be used as an assay, a detection method. And there's lots of work and interesting speculation on what type of assays can be designed. The basic Cas enzyme that we're using is Cas12a. It complexes with a synthetic guide RNA that has one sequence which is specific to the enzyme that binds it and forms a complex, and another portion which is complementary to the target. When it senses a target, it binds to it and then cleaves it, then dissociates from it like an enzyme action. But in this cleaving, in this very specific detection of the target, for our case, its target is DNA, double-stranded DNA, or it can also be single-stranded. When it detects and cleaves the target, the enzyme becomes activated, and we're showing that here by changing the color into blue to indicate the activated Cas12 enzyme. And the activated Cas12 enzyme indiscriminately now cuts single-stranded DNA. So in the assay, we include fluorophore quencher probes, and this then, the activated portion, once it detects the target, starts to cleave, producing a fluorescent signal, which we detect, and that's the assay. So to both do the purification up front and then to speed up the CRISPR-based assay at the end, we use isotophoresis. It's an electrokinetic focusing technique. We use a leading electrolyte, a high mobility anion, and a low mobility anion like a weak acid to electromigrate and focus target species flanked by a specific mobilities, leaving behind impurities. And again, we can use this both to purify. DNA has a relatively high mobility, so we can purify it. And then in the third step, I'll show you in the assay, we also use it to speed up these enzyme reactions. And those two sets of reactions, the first one is the target cleavage, activating the enzyme. The second set of reaction is the indiscriminate cleaving of quencher fluorophore pairs, producing the fluorescent signal. We first showed this in 2012 with secondary reactions that we can speed up by up to 14,000-fold by this very strong pre-concentration effect. We're using that now here for CRISPR enzyme. So just to summarize our assay, we have 2 on-chip steps and an off-chip step. So we start with a 5-minute extraction on chip using isotophoresis. We then pipette off, we go off chip and do a LAMP isothermal amplification. We first do reverse transcription of RNA to DNA, then isothermal LAMP preamplification or amplification. The main amplification happens here. That's the longest step of 20 minutes. We then dispense back into the same chip, but this time the channel is preloaded with guide RNA and the CRISPR chemistry, as well as the reporters. We speed up both reactions with ITP and detect it in about 5 minutes. These 2 steps are being shown here, but I'll show you some experimental data. On the top is purification, the bottom is the CRISPR assay. One of the first things we did is initially showed that the purification alone is compatible with a couple different downstream enzymatic amplifications, including qPCR. So this initial work was PCR. Now I'm going to show you the CRISPR. So this is now the third part of the assay. In this movie, we're switching back and forth between 2 color filters, a green color filter that shows the reporter cleaved DNA, and a red emission filter, which shows the labeled Cas12 with the guide RNA complex. So one of the discoveries of this work is actually that ITP can focus this Cas12. Somehow the gRNA speeds up the mobility, has enough charge, and lends enough solubility that it can speed it up and focus in ITP. This is also experimental data showing time versus distance. The straight lines indicate constant velocity and showing that they're approximately co-located, which is speeding up these reactions. Not only speeding up the target cleaving as well as the reporter cleaving, but once the reporters cleave, we also focus and localize, increasing the concentration of the reporter. So really, ITP serves at least 4 purposes here: the purification, speeding up the 2 set of enzyme reactions, and focusing the reporter. This is just further visualization. On the top channel here, we're showing, we've changed the grayscale to try to show you, but it's a negative result. So all you're seeing really is any degradation in reporters that result in a very weak background signal. And the bottom, the time is sped up, but it's shown here at the bottom of the movie. You see that within about 30 seconds, we can detect the target indirectly by detecting the reporter. And this is experimental data that's again shown here. In the assay, we detect 3 genes. So we do this in 3 channels: P gene, N gene, and the E gene. This RNase P gene is basically human detector to make sure the assay is working, and the N and E gene are the 2 COVID-19 virus genes that we went after. This particular assay is a demonstration of the entire process from beginning to end in this particular slide and this data. So here we squeezed all the time down to a 30-minute assay. So we spend 5 minutes on the extraction. We go off chip, do 20 minutes worth of reverse transcription and LAMP. Then we take that DNA amplicon. So initially we're focusing total nucleic acids, including the RNA. Now we go RT and LAMP. We take the DNA amplicon, dispense it into the chip, and this time we use ITP to speed up the CRISPR assay. And these data are showing positive patient data. On the top is 2 columns each, so at 1 minute or 5 minutes. This is data for the E gene, the N gene, and the RNase P gene. The bottom is a negative control where there's no target whatsoever. It's just viral transfer medium, and the reagents for CRISPR are downstream. So that just shows very small, faint trace of the ITP zone, which is probably from some small amount of degraded reporters. And these data show that for these dilutions that we undertook of this particular patient, we lost the signal at around the 3rd tenfold dilution. So it's part of our quantification of LOD, which I'll describe more. But we are able to do this all in 30 minutes. Lastly, here I'll show data from 64 human samples. On the left is data from COVID-19 positive patients. So for all of these 64 samples, we have qPCR data from Ben Pinsky's lab at Stanford. And what we show, the 3 columns here are the N gene, the E gene, and the RNase P gene detection result of the assay, of the on-chip CRISPR assay. This particular data that I'm showing here, we wanted to focus in just on the CRISPR assay. So for this particular slide, with large amounts of patient and healthy donor data, we're starting with pre-purified sample and then going right into the chip. So pre-purified with some standard porous column, and then going right into microchip and doing the fast CRISPR assay in less than 5 minutes. So the 3 columns are the N and E gene and the N RNase P gene. The negative healthy donors here are donors who tested negative for coronaviruses. They also tested negative for the flu. And of course, so we had no false positives. So our assay is very specific as per this data. In all cases, we're able to detect the the healthy donors' RNase P gene. And then Here, of the 32 COVID patients, we're able to detect COVID in 30 of them. The 2 false positives, we go back and look at the qPCR data, and in these 2 cases are the only cases where the CT values of the qPCR was 35 and above. So these 2 particular patients had the smallest trace amounts of the cohort of patients that we looked at. So with that, I'd like to summarize. We're, depending on which format of the assay we do it, we can detect from start to finish, from raw sample to answer, in between 30 to 40 minutes, depending on which version we do. Our limit of detection is equivalent to about 33, I'll report here about 33 CT. So it's in this range, equivalent to a qPCR with the WHO standard COVID-19 test. We demonstrated this on 64 human samples, including 32 COVID-19 positive patient samples. One of the things I'll point out is that this microfluidics approach, irrespective of which chip and how you use it, offers at least 100x lower reagent. In fact, for the data I showed here, we have about 10,000 times less reagent than typical CRISPR assays. And we have some interesting data for that, integrating the first 2 steps and the last 2 steps, and eventually want to integrate all 3 steps together. With that, I'd like to again acknowledge my collaborators, Ben Pinsky from the Stanford Med School, Niaz Banaei from the Stanford Med School. Of course, a special thanks to my student who led this work, Ashwin Ramachandran. Our funding is primarily from the Ford Motor Company. This is part of the Stanford Ford Alliance with program manager Mark Cabrales. We're very grateful for that funding. And also initial funding and seed funding for this came from part of a ChemH Institute, also at Stanford. Thank you.